propagating this technology in India. And you people should take it seriously and use it and produce it, or use or produce it. That is the aim of the main aim of my lecture, of motivating these faculty members and research students. Now, let me start with general introduction of Wikipedia. These are the following headings. I hope everybody is able to see the headings, right? Hello. The slide is clear to you? Hello?
Yes, sir. It is visible.
Okay. Okay. Now, coming to the discovery and abundance. Discovery is a historical thing, but still is important as far as we are concerned. The scientists should be recognized first, or any other thing. For that, I have thought that discovery is also very important. It was discovered by Harold Urey of America. He's an American scientist in 1931.
Hello.
You are not audible.
Sir is not audible. Yeah. Hello. Padmanabhan sir, you're not audible.
Sagar, can you just call sir and tell him?
I called sir and he's joining in just 1 minute. He's having some internet connectivity issues.
Oh, okay.
So he's joining back in 1 minute, he said.
Okay, no problem. Thank you, Sagar.
Hello. Just hello. Hello. Hello.
Hello,
Yes, sir, we
Hello.
can hear you.
You are able to hear me?
Yes, sir, we can hear you.
Okay, now the problem. Now, this again, I'm starting the discovery. You know that already I told about the Sarah Dure, this American Nobel laureate, and concentration, the abundance, coming to the abundance, seawater, 0.015% present in seawater. Why seawater? Seawater is a huge source of deuterium. And if you take the river water and general water, 125 to 150 ppm is the abundance. Even in our body, it's containing 4 to 5 gram of deuterium is present in our body. That is the natural abundance of deuterium. Coming to the production and availability, this is the most important thing that people are not aware of. Who is producing in our India? Only the production is done by the Heavy Water Board of Department of Atomic Energy. The deuterium, the largest producer, the highest purity in the world, we are only manufacturing. For your information, and people also should know this awareness about this particular product. Who want to really use it or you want to start it is very important information. And how it is done, then how much quantity is the produced in the thing. Is mainly mandate of the production of D2O is meant for the nuclear reactors. okay, as a moderator. I will come to next slides. I will be coming about moderator. And this process is how it is produced. Only the enrichment process. Hydrogen sulfide and water, natural water is drawn from the, especially in the this river water is drawn. So much river water is drawn and is getting exchanged with hydrogen sulfide, and this is enriched. After the enrichment, it is followed by vacuum distillation. They do by this two process. It's a very complex process. When you visit that, Managuru is one plant situated in Telangana state. If you see the thing, is a highly complex process. I can say it's an engineering marvel. The one the world such a class plant you cannot see in any other place. Wonderful place and wonderful plant they have got it. The highest the capacity is 150 to 180 tons per annum they produce it. They are mainly for the moderator purpose, that is nuclear reaction, that is nuclear material they are using it. Other than that, now currently they know the importance of non-nuclear application. They want to supply even the academic institutes. They want to encourage by giving the non-nuclear or propagating the non-nuclear application. They want to supply even some of the aided institute, government institute. They give free of charge, 1 liter free of charge. They give, and other private companies and private colleges they give charge. This is about the production thing. And this is very important to know the labeled compounds. As far as labeled compounds is there, you are supposed to know about certain terminologies. What is labeling? People think they don't know what is labeling. Labeling things they are putting the label. It's not labeling. Labeling means it is introduction of tritium atom into a molecule. It may be one atom or more atoms. Somehow you have to introduce in a molecule. You have to introduce tritium. This is also known as deuteriation or labeling. Both are same. But for that, how will you introduce the label or deuterium into a molecule? There are broad categories. I have divided three chemical broad class of reactions. One is chemical synthesis, another is exchange methods, third is biochemical methods, by which we can introduce deuterium into the molecule. Each why I have mentioned every reaction has got its merits and demerits or advantages or disadvantages. So when you take the chemical synthesis, there are substitutions, catalytic reduction using labeled reagents. These are the exchange methods and biochemical methods. What is the importance of one by one? We will see before this one more terminology we want to introduce: isotopic purity. It's very important terminology in the thing and deuterium labeling or for any other non-stable isotope labeling. Isotope purity is very important. Only with small example I would like to explain. In the end of the session, if any doubt is there, can clarify. Now, it is the percentage of specified isotopes at the given site. What is the given site? I have taken the molecule. Formic acid is the simplest molecule. I have taken formic acid. There are two hydrogen atoms. One is attached to right side, one is attached to oxygen, another is attached to hydrogen. Okay, that hydrogen attached to carbon atom is totally replaced by deuterium, 100%. That means isotope purity 100%. If it is 98%, purity means 98% of that particular position that given site, the specified isotope is replaced. That is the meaning of isotope purity. The isotope purity is playing a very vital role in all the non-nuclear applications. Isotopic purity should be more than 99% or 98 above percentage is very much required for this thing. For the detection as well as for the finding of the position. What is the very important is there are the when we see the thing that I will explain what is the method of detection thing that we'll see later. Now I hope it is clear that particular site, there are two sites out there. One site is totally enriched with that thing, other site is other OH is not at all. So it is the percentage of specified isotopes. In this case, specified is deuterium isotope, and it is occupying the carbon attached to hydrogen. That hydrogen is totally replaced. That is the thing. If it is 100% totally replaced, it is 100% isotopic purity. The chemical and exchange methods are there. Why chemical? There are different methods. Why it is required? What is the importance of the thing? I would like to explain here. Chemical method. When you take the chemical reactions, it will lead to specific labeling. I have given two examples. I hope you are able to see that only one deuterium is introduced in one benzene ring, and another is all the 6 hydrogen atoms are replaced with this. So what is the thing? In the specific labeling, if you want to particular position, people can use only chemical methods, not the exchange method. Exchange methods, most of the exchange methods are leading to random labeling. In which position, how much percentage is incorporated, we will not be certain. However, we can increase the percentage of isotope purity by stage wise. Once the exchange is done, once the equilibrium is reached, further exchange is not possible. But again, you take the product, again label it subject to labeling, then it can happen that 100% purity can be obtained in exchange process. In the exchange process, what they do, they take the compound to be labeled, and D2O is taken, and catalyst is taken, and the reaction is done in the closed system. That is catalyst, compound, and D2O. That is the raw material we are using it. With this, we can exchange the compound to be labeled. This is where the exchange label is done. Exchange label is not only for tracer purposes useful. At same specific site, that the chemical labeling is leading to specific site labeling. That is for the reaction mechanism, enzymatic reaction. What is happening to the site, the fate of the molecule can be studied. That is the use of chemical and exchange methods. Now coming to now clearly catalytic reduction. Two examples are given. Now you will understand the position, exact position. This is a totally chemical method. What we are doing, we have taken the one ester. The first case, are you able to see it? Hello? Hello? Hello?
Yes, sir. Yes, sir.
Yes,
We are
sir, we
able to
can
see.
see.
Yeah, okay. That first example is one ester containing long chain ester containing 2 double bonds. So in this case, we are doing two reactions. One is deuterium. Deuteriation, that is in the presence of deuterium, we are doing the reduction. In the presence of the catalyst, that is known as catalytic reduction. Platinum oxide is taken, deuterium is taken. What is happening during the thing? D2O is formed, inside to D2O is formed. That is responsible for the reduction purpose. Deuterium gas also will be there. Here you see that clearly you see that deuterium is occupying wherever the double bond is, that is getting saturated with deuterium. So specific positions it is getting there. And second, this thing, the ester is converted to second reaction is saponification or ester hydrolysis. We are doing it. Corresponding acid we are getting it. So in this case, we can make the ester to acid containing the particular position labeling can be. And coming to hello, hello, is it hello?
Yes, sir. Yes, sir.
Uh,
We can hear
okay,
you, sir.
okay. And second example is triple bond containing a compound we have taken. In this case, the triple bond is deuterium, same catalytic reaction we are carrying out in the presence of ethyl acetate. When you do the reaction, exactly the position is particular position only it goes. Most of the reactions, this type of catalytic reaction, give rise to the position labeling, exact position labeling. Some cases, the scrambling happens. Otherwise, most of the cases, it goes to particular specific position. So this is very much useful for the study of what happens to a certain particular site, what is happening during the enzymatic reaction. Some particular site is affected. So that site specific and reaction is studied. Now coming to another example also will give you reduction using labeled reagents. There are two reactions are shown. One is the ordinary reaction with using hydrogen, sodium borohydride. All of you know this good reducing agent. So we can use sodium borohydride reduction for the acetophenol is reduced to the corresponding alcohol is shown. But instead of sodium borohydride, sodium borodityrate is also can be used. Okay, but we will get the deuterium labeled thing, and this position we are getting it. Particular position we are getting, it is not at all labeled in the ring. So if you want to control the thing, you can use that because the reaction mechanism, one should know that borohydride always a hydrogen ion mechanism goes to the carbonyl group and activates the carbonyl group. So when you know the reaction mechanism, one can implement where it should be labeled. You want the ring label or side chain label, that depends on the user's requirement. We can use the reagent, we can do the carry out the reactions. This is the advantage of it. And different labeled reagents are there. It's very important. Lithium aluminum hydride, that we can label it. Lithium borohydride we can label it. Even for methylation reaction, methanol also we can label, or methyl iodide also we can label it. So this is the use of labeled reagent which will go to the particular site of labeling can be. And biochemical methods, I have not taken the deuterium thing. I have taken L-alanine. I have taken but the simplest amino acid, simple amino acid next to glycine, alanine is the next member. If you take the L-alanine, when you oxidize with L-amino acid oxidase, the enzyme if you are using it, what happens it is converting into pyruvic acid. Wherever the enzyme reactions are applied to hydrogen molecule, the same can be applied to things for pyruvic. You know, reductase, oxidase, kinase. Many enzymes are used for the production of deuterium liquid. So coming to this one, detection and toxicity. The first part is if you label the organic compounds, all the compounds are contained this. We are replacing with the deuterium label we have prepared. And if you don't have the detector system or quantification system, there is no meaning in doing this experiment, or all the whole effort will be wasted. Thanks to the advent of mass spectrometer and NMR, now the mass spectrometer LCMS coupled with the LCMS are becoming powerful. These things are now available with many industries. Because of that thing, we can quantify in very low amount of deuterium labeling tracer work. We can quantify it. At the same time, NMR also useful for the position of the inorganic chemists generally use mass spectrometer for the NMR spectroscopy for the elucidation of structures. Using the deuterium also, one can position which position it has gone and how much is quantified that the isotopicity is obtained. Everything can be analyzed with the NMR as well as with the mass spectroscopy. And previously, the history talks about that. Before that, what we were using is why we are talking about NMR and mass powerful spectroscopy, because previously the threat was supplying the C14 and tritium, which are radioactive in nature. What was the problem? The thing is, it's also sensitive and everything is same. The isotope of hydrogen, tritium is also isotope of hydrogen. The chemical reactions are same. What is the problem with the detection part is, here you require the level, this liquid simulation counter is required, or NMR and mass spectrometry. Nobody is allowing the thing because C14 is having the half-life of 5,700 years and tritium is having 12.5 years. So when you are doing the tritiated compound or C14 compound, nobody was allowing the thing to have using the mass spectroscopy and NMR. And people used to tedious work like chemical degradation methods they are using it. Now because of the invention of this LCMS and NMR, powerful NMR, and people use stable isotopes are very much useful. And now that totally it is phased out. Tritium and C4, previously we were supplying to some companies. Now it is totally we are stopping. We are almost stopped because the domination of deuterium and C13 has come in the picture. Okay. That is the importance of deuterium. So toxicity. Coming to toxicity, very very important factor one should know before handling. As a heavy water, if it is toxic, if the more than 25% of the body fluid is replaced. The body fluid, you know that around 60 to 70% is water. Our body contains that 60 to 70% water. If out of that, 25% is replaced, then it becomes toxic. Otherwise, there is no toxic for less than 10 because even for clinical trials and some of the nutritional studies, people are taking some ml of D2O. Pure D2O is taken and people are already using it. That's no problem. But they are not reporting anything. And only that more than 25% body fluid is replaced, then it becomes toxic. Now, as such, there is no safety precautions are required for handling that D2O as a D2O. Then coming to the deuterium gas, what deuterium gas is not at all it's not having toxicity because deuterium gas, when it is inhaled by humans, first of all, the exchange rate is very, very less. It is not exchanging the body fluid with water. So, for the water, because immediately we are inhaling and exhaling, the process is very short. The short period without catalysis, the exchange is not possible. For anything to think some amount of catalyst, acid catalyst or base catalyst or real catalyst are required in our body system. That is because the short time of exposure, it is not getting exchanged with water, and hence deuterium gas is not.
Hello, sir. May I interrupt you for a while?
Yeah.
Sir, our audience are not able to hear you properly. May I request you to just keep the audio, Miss microphone, near your mouth so that just close to your so that the voice is clear. We are not
Okay.
in a position to hear you clearly.
Oh,
Thank you, sir.
oh my goodness! You could have told me earlier, no? Now we have now
Uh,
it is audible. Now it is audible. Hello.
yes, sir. Yes, sir. Yes, sir.
Now it is okay. You please, whenever the problem is coming, please inform me so that it should be a main purpose as to everybody should hear it. That's
Yes, sir. Yes, sir.
now it is
Thank you,
clear.
sir.
Now it is clear.
Yes, sir.
Okay. Now it's
Yes,
okay.
sir.
Okay. Now, if you want me to repeat the previous slides, you can tell me the thing. I can clarify the things. If anybody is having the thing, I can tell. Okay. Now, again,
No
go ahead.
worries, sir. I think we
Now,
will continue. Yeah.
shall I continue with this?
Yes, sir.
Okay. Any problem in that, please interrupt me. Okay.
Sure, sir.
Okay. Now, precaution. When coming to the precaution, what are the precautions you are supposed to take for the handling of the thing? I told you that there is no precaution required for as such while handling the thing. Only drinking should not be there. And otherwise, that tritium handling is very, very safe and people are using it so many years. In abroad, so much people are using it. India now started using it. Without much problem, they are using it. But D2 handling tons and quantities are used by heavy water boat. There is no report of problem with the heavy water. Only thing is that drinking, I told you, we have to be very careful. People should not misuse the water, that they should not mix up with the water. That is the only precaution we have to take. By mistake, there should not, water bottle should not be exchanged with it. That type of precaution only take. Okay. Now, what are the precaution for? I told you that isotope purity is very, very important. That in the all cases of deuterium labeling, I told you during deuterium labeling, therefore, we are taking so much precaution. During labeling, techniques are only techniques are important. Gaseous reaction, when you do the gaseous reaction, suppose catalytic reaction, if you want to do it, in presence of deuterium gas, all the things should be handled in the vacuum line setup. You have to use it, or if you want to use it, moisture-free atmosphere with the total with the moisture that the trap should be attached and the balloon atmosphere or dry box conditions. We can use the reactions. We can handle the reaction. The transfer of heavy water, one should be very careful. The transferring also, right from the transferring, one has to be very careful. We cannot just like the transfer. The open atmosphere contains the moisture that will immediately will exchange with water, and the percentage of radioisotope purity of water will go down. When it goes down, there is no meaning in doing the thing. Yeah. Now, what is the thing? What is important for isotopic purity is it should be one drawback with the system is all the different compounds should have more than 99% isotopic purity. That is, the particular site it should be with 99% of the deuterium should be present in the particular site. 1% the hydrogen is tolerable, it is okay. Why? Because why the high purity is required? That instrumentation like bar spectrometer and NMR spectroscopy, that interference of proton will come in this case. So the proton will give the signal that the interpretation will not be proper. And once the background is increasing, sensitivity will go down in any analytical systems. So considering these points, so people should be very careful in handling doing the reaction. They have to be very careful. Okay, even right from the transferring of the thing, but once it is diluted, there is no meaning. I told you actually. So we have to be very careful doing that. Now, application. Coming to the now, we are coming to the application part. There are two broad classifications: nuclear applications and non-nuclear applications are there. This, in the nuclear application, I know you know that there are four applications. Only I have mentioned four applications. That limited applications are there: coolant and moderator in nuclear reactors, the fusion reaction, deuterator precursor for the F-18 molecules, and production of isotopes inside the. Now, coming to the nuclear application, that is our mandate of heavy water board. Heavy water board is manufacturing highest security and the largest quantity of deuterium in the world. They are producing 180 tons, 150 to 180 tons are produced in Bangalore plant. And so many plants are there. They are all very little smaller plants. They are also producing heavy water. In Alibag is also there. Rajasthan quota is there. Like 6 plants are there, out of which 3 to 4 are the operational. So, our requirement is for the reactors, wherever the natural uranium is used. I'm not going into the detail of this, just outline I'm giving where the nuclear reactor is used and why it is used. It is used as a coolant and moderator. Moderator is a very important material in nuclear reactor where the natural uranium is What is moderator? Actually, the neutrons are coming with very high energy to moderate the thing. That is slow down or reducing its energy is very, very important. The fission reaction. To sustain the fission reaction, this neutron energy should be reduced. So why we are using the deteriorated water for reduction of energy in the thermal, and we bring the energy to thermal neutron. Thermal neutron are having the energy 0.2 0.025 electron volts are there. That much energy, very low energy it is. So that much energy is reduced from million electrons to this much energy. That working, the moderating capacity of DTO is very high. Therefore, it works as a moderator. This is the major use of this moderator in the nuclear reactor in India. Okay. But it is also used as a coolant. Coolant is the main purpose of coolant is just like water. It is heat transfer. It is wherever the core reactor, nuclear reactor, the core heat energy is produced. To remove the heat energy, heat transfer, coolant is also used.
Hello, sir.
Yeah, tell me. Hello.
Sir, your
Yeah.
voice is still breaking. We've been receiving many chat messages saying that the voice is still breaking. Would you please be louder so that
Okay.
so that there's
Sure,
a
sure.
clarity in the voice?
Okay,
Thank you,
okay,
sir. Thank you. So
okay. No
sorry,
problem.
sorry for
Yeah, no,
disturbing.
you please, because of the systems are not so much perfect in India. There's a problem with the thing. Okay, it's not
Thank you,
standardized.
sir. Thank you.
Okay, anyway, that you please it should be audible to everybody. There's some people are telling something that is not. I'm not very happy with this. Anyway, hello.
Yeah, just be louder,
Now,
sir. I think it will be better.
okay. Now let's fix the thing. What? Just a minute. Huh? Hello. Not yeah?
Yes, sir.
Hello.
Yes,
Now
sir.
I go ahead. Now, coming to the fusion reaction, I am not going to talk about the fusion reaction. Fusion is the basic reaction is based on bomb. So deuterium is also can be used in the fusion reaction. So that is not our topic. Now, coming to the deuterated precursor, about the F18. F18 is a positron emitting radioisotope, which is used in nuclear medicine. Hello. Hello. Some
Yes, sir.
disturbance. You are able to hear? Hello. Is clear?
Yes, sir. Yes, sir.
F18 is a positron emitter. It is used in the nuclear medicine mainly. It is the positron emitter is mainly used for the imaging of the organs. Whatever the effects of the even cancer diseases or any diseases, the diagnosis of cancer, it is mainly used. F18 radiopharmaceuticals. In this case, some of the molecules, we want to image the particular organ. That some of the compounds they eliminate or they clear faster from that particular organs. Those medicinal chemists thought that by replacing the hydrogen with the deuterium, that is the one way of approach to delay the process. Once the delay is happening, because of the the deuterium is slightly slower kinetics, because of the slower kinetics, so they can delay the process for imaging so that particular precursor will remain in the particular organ for some more time so they can image. This is particularly for the imaging purpose. Their deuteration work is going on. Almost they are successful. In future, deuterated precursor will be useful in the nuclear medicine, especially in the F18 radiopharmaceuticals. Now, coming to the production of isotope, this you would have studied, most of you would have studied in college time. The cyclotron is the accelerator, the particles are accelerated and bombarded the target material to get the particular radioisotope. So that also, the deuterium is particle accelerator used and some certain isotopes are produced in the cyclotron. That's all about the thing that the main nuclear application is in India is largely used is moderator. That's the thing. Now coming to non-nuclear application, there are many fields. I have not included certain fields of there. It is a vast field, but I have restricted to material science, animal solving, food, cosmetics, agriculture, life science, pharmaceutical, and others. Okay, so many fields are there. This huge field that it is not possible to cover all the fields. So I have restricted to some fields which are very important and practically useful. Now we are using the for non-nuclear application, what is the property we are exploiting for the non-nuclear application? Most of the non-nuclear applications, we are using the deuterated compound. Why? Because this is the fundamental thing one should know. CD bond is stronger than CH bond. And the cleavage of CD bond requires greater energy than CH bond cleavage. This is the fundamental property. It is responsible for usage in the non-nuclear application. Okay, this property is only exploited in all the non-nuclear applications. Now, coming to the thing, why it is stronger? Because of 0-point energy. So you see that the 0-point energy of the carbon to CD bond is having lower 0-point energy than CH bond. If you see that because of that 0-point energy, it results in a lower ZP, results in a higher activation and slower rate of cleavage of the CD bond. This is the fundamental thing, reason for why the CD bond is stronger. In the other language, we can say is the short bond length. It is shorter than the CH bond. So when the proximity is closer, the bond strength is increasing. Now, we are always talking about CH bond. What about other bonds? The organic chemicals, organic compounds are containing NH bond, OH bond, SH bond. These bonds, why we are not talking? Because they are called exchangeable bonds. You will see that the hydrogen is attached to nitrogen, oxygen, sulfur. They are all electronegative atoms. Nitrogen, oxygen, sulfur, they are all electronegative atoms, more electronegative than carbon. So when the hydrogen bond is attached to nitrogen, the electron pull will be towards nitrogen, electron pull will be towards oxygen, electron pull towards the sulfur because of electronegativity. It pulls towards it. So it can be, the H can be released as H plus ion easily. That is known as nearby. That means if you are exchanging with D2O, this nitrogen bond containing amino acid or some alcohol, SH bond methionine, some type of amino acid, easily it can be exchanged. At the same time, once it is labeled, it will become ND bond, OD bond, corresponding LD bond will be formed. But what is the use of that? Then again, that if you are changing a hydroxylic solvent, if you treat it again with water or ethanol or methanol hydroxylic solvent, it will go back to original to NHOH, SH. So there is no meaning in labeling the things. Therefore, we are always talking about the CH bond. But however, in protein studies, certain studies this replacement of nitrogen, oxygen, and sulfur are used in certain cases, not in all the cases. When this material is stored in the deuterium thing, then the exchange will take place. That stabilization studies is useful. Otherwise, there is no much useful. And coming to material science, the first application of material science you take it. That you must have seen that fiber optics, that people, commercial people are more intelligent. They immediately exploit the thing wherever possible. They go for the cheaper thing and they understand the what is the technology and immediately they implement it. Fiber optics is a very good field and what is it, very highly commercially field. They are the optical fibers. They are using it. They are made up of silicon. SiO bond is there. That is made up of silica. But when they use it, that they are getting degraded faster. And signal, they come to know that signals are also transmission losses happening so much. Then they thought that why not we apply deuterium? Deuterium, when they apply with the thing that already they have done it, that was quite successful. That the SiOH bond is replaced by SiOD bond. That the lifespan is that the shelf life is increasing, the lifespan is increasing. At the same time, the particular OD bond, it is not absorbing the wherever the transmitting the particular wavelength is not interfering with the particular OD bond. Therefore, there is no much transmission loss in the case of optical fibers. So the optical fiber technology is always using it. The Sterlite, one company in Sterlite, they were using the deuterium for the labeling of optical fibers in the industry. They were supplying, the Hayward board was supplying that deuterium gas during that some years ago. Now coming to the organic LED, that organic light emitting diodes, they also use the thing. It's the same principle. CH bond is stronger than CD bond, so the lifespan is increasing for this thing. In addition to this thing, I included the UV lamps and spectrophotometers. This is a very interesting field. I came to know that here engineers and different audience are there, engineers and chemists and pharmacists are the main audience I heard. So what is the very important thing I want to highlight? UV lamps, spectrophotometers, thousands of spectrophotometers are there. Till date, nobody is manufacturing as per my knowledge. Most of the UV lamps are imported. The UV lamps containing deuterium discharge lamp is used for the UV detection, UV range. The lamps are used. And all thousands of spectrophotometers one can take this particular. So think about it. The manufacturing of UV lamps one can undertake. Because when we see the Internet of Thing in the BRC, we are seeing so much UV lamps are being imported from Germany and US. And coming to the HPLC, of the thousands of HPLCs are used by pharma sectors and R&D sectors in India. That also, these two fields are untouched fields that one can easily. It's not a very big technology to make the UV lamps. A lot of the companies are the expertise is available for making lamps, but one can think about it, make the UV lamps. That one area of scope is they are using UV. Another area is NMR solvents. It's a very big area where the solvents are, whether that deuterated solvents are routinely used and widely used in NMR spectroscopy in India. Most of the pharmacists, research institutes, and many R&D labs are using this particular solvent for the elucidation of structures. All the reaction, even for reaction monitoring, even day-to-day reaction monitoring, people are using the NMR spectroscopy for monitoring of the reaction of the reagent. So these are the main solvents: chloroform, dimethyl, sulfoxide, acetone, acetonitrile, benzene, methanol. These are the D means only one hydrogen is replaced by deuterium. D6 means there are 6 atoms of uh, deuterium. Then 3 acetonate 3 means 3 atom. So number indicates the number of atoms wherever totally replaced. So this compounds, these solvents are manufactured in huge quantities. They are supplied. They are now we have started around 2000, around 10 years back. We have initiated the VRC, that is VA has initiated, and they are now started manufacturing this of the solvents, and they are supplying. That British marketing these products. So anybody wants to do this thing, I want to give the information. This, the following solvents are available except methanol. Methanol is not available with Brit. Most of the other solvents are available with high purity. One can use it with it. When compared with the Sigma is supplying, Emerk is supplying, some other foreign company, Emerk, other companies are supplying, they are costly. When compared with thing, even for research institutes, that the GST is less for this thing. So one can procure from Britain. That is my request for you. Coming to, and in addition to that previous slide, this utility, in addition to the use of NMR solvent, that also that the example methanol also can be used for labeling of the molecule. Methanol, acetone, nitrogen, even chloroform is also used for labeling of the very important compound they are using in labeling also. Even benzene related compound, starting can be benzene T6. Methanol, you can use it for methyl iodide preparation. Methanol is a very important reagent. Important starting for methyl iodide. Methyl iodide, that is CDGI, is very important for the methylation reaction. So, coming to the food, that not much slides of the food that not much heavily used. It's just started and people are using it, but not much is reported. Heavy water used. This is the study they carried out at IAE, International Atomic Agency in Vienna. They have got the program for nutrition studies for lactating mothers. They have studied the thing, how the nutrition taken by the infants. They have studied it. So this is the one just report. In addition to that, they are trying to analysis part also. They are using the water food analysis. That's a tracking of how much is any contamination is and all the things. There various types of analysis they are using the water. It is not much reported I can say. Now coming to cosmetics area, what is very important that second lipids that it's affected by oxidation. They want to strengthen the lipid, reinforce the skin cells using the deteriorated products. The main thing that recent study showed that oil penetration studies. Previously they were doing with deteriorated water. Now they are started with, because Hindustan Liver supplied some amount of appreciate the water for the studies along, but the thing I'm telling, they have studied the skin penetration and oil penetration studies. They were using it. Even the cream also, they are trying to manufacture containing material so that it can strengthen the thing. In the anti-aging cream, they call it as they are studies are going on. So cosmetics also, they would like to use this material level. Coming to agriculture sector, in agriculture, it is now previously they were using C14 and Trishul, they were using it because I told you, again, because of detection system and the expertise is available because of the labeled compounds are available of labeled compound, available of expertise and available of instrumentation. This isotope, radioisotope technology totally replaced in agriculture sector also. What is this? Previously they were taking, they are studying the environment studies. What is environment? So much, you know, that in agriculture, so much pollution is happening because of dumping of herbicide, pesticides, so much spray, so much tons and tons quantity are used by this agrochemical companies, manufacturing companies. So it's a total burden to the soil. So in the soil, what are the types of, how much time it is residual, this agrochemicals are present, how it is affecting the animals, how we are, because we are ultimately we are also eating animals. Somehow it enters to the animals. People may not eat it, but through the milk also it can enter. So what is the effect of humans? What is the effect of animals? What is the effect? How much is residue present in the plants? How much is reduced present in the, how long it is present in the soil? All the things can be studied using this best technology, mainly in agriculture. Agriculture also, still I can say this type of medicine type of thing, pharmaceutical sector type. It is a medicine for the pesticide or the herbicide or used as a type of medicine for the plants, I can say. Therefore, absorption, distribution, metabolism, excretion, that is admit studies, toxicity studies also. They have to study and submit to the safety assessment to the agrochemical board. Then they will, for the registration of this, then only the registration can be done. So it is similar to your, I can say, pharmaceuticals. So petroleum life sciences is a huge thing. Only certain things we are going to talk about. Thing. High water, they have already high water board already done research on particular stabilization of oral polio vaccine. Is there that they have studied some long back. They have studied it, but the study was quite successful. But due to some reasons, they cannot come up with the result that they cannot implement the thing. What is the use of that oral polio thing when it is transferred to remote villages? Hello,
Hello,
it's
sir.
transferred. Ah,
Hello,
can
sir.
you hear? Yeah.
Sir, voice is now breaking. It was
Okay.
clear so far. So just be louder. Thank you.
Okay. Now. Okay. I'm. Hello. Now it is clear. Hello.
Yes, sir. Yes, sir.
Okay.
Yes, sir. For me, it is clear. Otherwise,
Yeah,
also, but
but
a few of the
okay.
participants are not able to hear. So just be
Fine.
louder, sir. Thank you, sir.
Okay. No, I am. No, any problem with the audience only is very important because we are
Yes, sir.
still.
Yes, sir, indeed.
Okay.
Yeah.
Now, heavy water. I told you that polio, oral polio vaccine. The heavy water board has studied the studies, conducted the study. They were successful because of they were telling the because of medical lobby. They could not implement this particular studies in the implementation of oral polio because oral polio when it is transferred to remote villages, what happens? The remote villages do not have the refrigeration or cooling facilities. There, that when you are storing this vaccine in the D2 or the heavy water, it is getting stabilized. That nothing is affecting. The safety is maintained. Whereas in the other vaccine, if you are some other water uh, medium is there, one has to freeze it. That the stabilization is very poor. So the stabilization of the biomolecules is very much helpful. That people already studied it. However, the enhancement of the thermal shock, the thing is utilized in other vaccine, cattle vaccine, they are using it because there is the much not much regulations are required. So people have started with their vaccines. So one can think about now COVID-19 also. If the vaccine is coming out with successful, one can think about the stabilization using DPO. One the pharmacist should take it seriously and implement this type of activity wherever possible. We have to study the thing and come out with good results. I hope this will also be helpful. Now, coming to the biotechnological use. Biotechnologically is very, very important water because in our Brit, what we are using, that very important thing I want to share with you, L-amino acids, that is carbon 14 labeled, that is radioactive amino acids, we are producing, mimicking the photosynthesis of algae. Algae, blue-green algae is taken, and we are conducting the photosynthesis inside the lab with particular elimination, and we with the water and C14, the carbon dioxide atmosphere, carbon dioxide will be C14. So all the C14 will be incorporated in the algae, and when you hydrolyze the harvested, when you hydrolyze the protein, you are getting the amino acids. What is the specialty of the amino acid preparation? One cannot prepare even by chemical synthesis. It will take hundreds of years to make all the amino acids because that L amino acids are only important biological system. L amino acids are present. For study also, we require L amino acids. But when you do the chemical synthesis, very tough job, and rationalization always occurs against the separation. So much loss is there, so much very difficult to prepare by this method. This is the method people of biochemistry have exploited, and they were successful. Long back, the technology is established in our grid, and we have been supplying amino acids for different research institutes, including Freeman Institute. We have been supplying long back. Now it is almost stopped. The same thing, line. One can think about the incorporation of deuterium into the amino acid using this method. The technology is available. Only one has to try and succeed because many reports are available. The other people are here. When the abroad people are able to manufacture or synthesize, why we cannot? It is very easy to synthesize. Only thing is focusing is very important. Dedication is important in this area. And one can talk about how. The question is how 100% D2O is stable. That the algae and bacteria stable in the sense that they can survive or not. How they are reaching to 100? What the thing is, they are initially they grow in the algae and bacteria 20% D2O abundance. Then they stepwise they increase into 20%, 30%, 40%. They increase to 100%. Like that, algae this thing is increased. That adaptability is increased. Like that, one can think about it. Even the biochemicals like sugars and carbohydrates also can be made. We have got technology with this that we are ready to transfer it. If you collaborate with Brit, that is also possible for your studies. Glucose, fructose, all things can be synthesized using biochemical methods. Now, coming to hello and probing of hello. Hello? Hello?
Yes,
Hello?
sir.
Yeah,
Yes,
somebody's
sir.
Right. Now,
some.
now some disturbance is there. That's what just I thought. Anything is clear?
Yes, sir.
Hello?
Yes,
Okay. Voice is clear, no?
yes,
Hello?
sir.
Because
Yes, sir.
here some disturbance I mean. That's why I just interrupted. Sorry.
Yes,
No.
yes.
Probing of biological phenomena. D2O provides unique avenues for exploring basic mechanism of biological phenomena we have written, and alpha amino acids are also widely used for mechanics studies. What is the special unique avenues mean? Only that hydrogen bonding is available with OH and SH and NH. Then what is happening to the thing? Immediately that they label it and one can study using the uh, deuterium. Whereas other isotope, oxygen isotope, we have to label the oxygen, nitrogen, we have to label it. In the case of sulfur, also one has to label different isotope. Whereas in the hydrogen, in the one isotope, we can we can explore the basic mechanism containing OH bond, that is NH bond, SH bond. This is the specialty of the thing, in addition to CH bonds, that is CD bonds. Now, protein research. If you see the protein research papers, really you will really astonished. So much work has been done using the deuterated protein conformation and protein studies, protein shapes, and protein synthesis. To tell anything on the protein, people have used. They have used enormously. They have used the deuterium in this work. So protein function is written by that the conformation. With the conformation study, what they do, the hydrogen deuterium exchange method, mass spectrometry is proved very important. If you go through the papers, you'll come to know a lot of work is done in the thing using deuterium. Now, nowadays currently that the protein, lipid, and nucleic acids also labeled with the deuterium, they also are used in the presence of some other substrates are studied in presence of some other compounds. They are studied accurate measurement, dynamic measurement, how the movement is taking place, how the shape is changing. All things can be studied in presence of some other chemicals also. Now, coming to fatty acid research, it is a pharma people will be knowing, but better polyunsaturated fatty acids. I have written saturated acid in the fatty acids are essential nutrients for everything the brain function to cell function. The brain is rich of polyunsaturated fatty acid. Okay. So that thing is now it is getting research is very good research is going on in this field because you will come to know what is the applicable in pharmacy. You will come to the next slide. I told you that the brain function to cell function is very much useful and it can be supplemented with outside food. We can give the polyunsaturated acids. What the chemists have thought about it, since Alzheimer disease and Parkinson's disease are oxidative, tied to oxidative stress, due to that thing, these are responses, brain function and I told the cell function, they are all uh, uh, easy to getting easily oxidized, then they are under oxidation. This diseases are also forming due to that. That's what they predicted. So what they can do, there are two approaches. One is giving the antioxidant for repairing. Otherwise, use polyunsaturated fatty acid. One can strengthen it by substituting the hydrogen atom with deuterium. The second approach, they found that it is really better and it's giving good results. Now, breakthrough has come in this case. So once the breakthrough is really come out with good results, then this can be that Parkinson, whichever the oxidation, due to the oxidative stress, what are the diseases that one can easily battle with the substitute polyunsaturated fatty acid with deuterium. Now, coming to next one is not a in the life sciences I get it because total body water people are using the tritium that is using the isotope dilution method. Now, D2O is very much useful for tracking the fluid changes between dialysis. What is between dialysis, pro-dialysis, and post-dialysis, pre-dialysis and post-dialysis? The doctors they study the fluid changes. That is very important, clinically very important. That valuable information they are getting it. So before getting they analyze the water content or the fluid, what is happening, and after that, what is happening, they are very much useful in the improving the dialysis management. This is the recent study, and of course they have studied long back, but this is also very important, gaining importance in this field. Now coming to the your field, I hope that many pharmaceutical chemists are there. I would like to talk about only the pharmaceuticals. That all of you know there are many uses are there. Only what my interest is, your interest is one, and my interest is also there. My question is standards in mass spectroscopy, biochemical study, biochemical studies, tracer study. Major use in drug discovery process. These are things are involved, and drug development process are very much important. When I because this when I practically visited ran vaccine to Glenmark to many companies I visited during my service because we wanted to start the deuterium labeled compound since I was having the experience with tritium labeling of compounds. I thought we can take it up this one. When we then so much quantities of that's huge quantities are being imported by different companies. Around 50, roughly 50 odd companies are there who are involved in clinical uh, trials. So those who are the patients are there, they take out the patient's blood sample, they analyze this thing, mass spectroscopy, they are having LCM, they are analyzing them. Uh, Drug content using the deuterium labeled standards. In the standard, isotopic dilution techniques are used for finding out the concentration of each drug and metabolites also used. So therefore, I thought this area is a very important one. In standard mass spectrometry, stable isotopes, that's very much used by the industries in pharmaceutical sector, mainly the clinical trials who are conducting many big laboratories are taking up the thing. So large amount of compounds are being regularly used, and the worth is you can say even in a year 2 to 3 crores. Even small laboratory. I recently I visited some laboratory just for my information. very small. I don't name the thing. I'm not sponsoring that level, but they told that 2 to 3 crores worth of materials they are taking this in Mumbai only. They are taking deuterium labeled compound. In one company, 2 crore. You can think about 50 companies, 100, even not even 100 crores, even let it be 10 crores. No one can talk about, but 10 crores out of which 90% of being important. One or two companies, when we initiated, some 2 companies from Bangalore, they are doing that work and they are supplying in very small scale. In Bangalore and Hyderabad, 2 companies are only involved. My request is pharmacists or chemists or think one should look into this particular area of deuterium labeling, and you can also start your own way. That's what any support is there. I am ready to get the help for you. That is mine. In bioavailability studies, I need not explain the bioavailability studies. The thing, the bioavailability is more, the drug is more available, or this optimum bioavailability there is always good. Some of the drugs, they people screen it for the what are the drugs are available with the low bioavailability. They want to increase it by what is the approach they are approaching. Now the good approach is to introduce deuterium. When you introduce deuterium, the bioavailability increases. And all of you know because CD bond is stronger than the CH bond. So in most of the cases, bioavailability increases. So these studies are regularly studied by pharma industry. Coming to bioequivalent studies, the bottleneck is bioequivalent, bioavailability, and toxicity studies are very important bottlenecks in our pharmaceutical industries. These are regularly studied, the radioisotopes as well as stable isotopes, because of the increasing number of instrumentation in India. Now they have started only stable isotopes are dominating bioequivalence. They want to study that bioequivalence. They take the 2 drugs, whichever is cheaper one they want to study. Then if it is cheaper than the corresponding drug, then they will apply in India, then they will sell it, they get registered for it. That is the main purpose of doing the bioequivalence studies, I think. Now coming to tracer studies, absorption, distribution, and metabolism studies. This is a very important drug development. Previously, C14 was dominant. C14 and tritium level compounds were using it. Now, currently, D2 is almost replacing the radioactive isotopes. But it was also very much useful in the mainly for the quantitative assessment of some drug, mainly the liquid medication. They are very much. Now, one question will come to your mind: deuterium incorporation, whether solve all the problems, all the inter APIs if you are using the labeling it, whether we can solve the problem. No, people have already reported all the compounds when you label it with deuterium, it will not serve the purpose. Okay, and every position is useful. No, that also we need to study a judicious choice of both the molecule and site of action must be undertaken. What is judicious thing? We should know the reaction mechanism where it is going. That's when it is affecting that the enzyme is affecting which positions affecting. We should know. At the same time, deuterium incorporation also we should do it because when you are going on exchanging with all the atoms, then the drug will become costly affair because we require more deuterium. Deuterium is also costly, so you can judiciously one has to use it both the case of knowledge as well as the practical thing working metabolic rate where it is going, or all the knowledge should be there to select the position. Now, due to the high biological half-life, the biological half-life is increasing. It's the advantage for in one way is that the potency is increasing with small, longer time, and sustained release in the body also. The sustained release is there because the CH bond is once it replaced with CD bond, kinetics changes in 6 to 10 times it is lower. So slowly that is degrading. That at same time bioavailability is available and the potency is not affected, and it is slowly decreasing and the same effect is attained. Coming to the next one is it can improve pharmacokinetic and toxicological properties due to stronger carbon modifying the metabolism. In this case, most of the cases, toxicology properties are also coming down. Now, this. Now coming to the next one is deutetrabenazine. It's the recent, that is first deuterium drug to be approved by FDA, US FDA. 2017 it is approved. Its name is due tetrabenazine. It is the deuterium version of tetrabenazine. You know, this very much used long back as a, with hydrogen. It is used as a Huntington disease. It is very much useful. What is the benefit of this thing? When we label a particular position, it is giving the long of a time, long half-life, so that bioavailability increases. So the benefits are more. So they thought that it can implement this. It can give much beneficially effects than ordinary hydrogen tetrabenazine. Now, this is the structure of the thing. You know, the structure. When you see the structure, where they are labeled, you see only oxygen. That the oxygen is attached to methyl position. Methyl group is attached to two oxygen. That position will be labeled. Why not the other position? Why not so many positions of the ring labeling? You have not done this thing. You know, they have studied with limited amount of this. The function they have studied. It is effective. When you are labeling with more thing and the effective same, then with less amount of material also the effective same, people will go for less labeling. So in that way, selective positions are very important to label. All positions we need not label it. And if it is effective, one can implement one position. One methyl position. If they label it, they may not be much useful. Then they have come to the two oxygen position methyl labeling has been done. Then they found that it's very quite useful. Therefore, they used it. Finally, they arrive at the thing. A lot of R&D has been done and come to the finally with limited position labeling is helpful. You know the advantages of things. I need not discuss all the things. Bioavailability increases because of slower kinetics. Kinetic isotope effective. What is kinetic isotope effective? Like I tell you, when the kinetic is because the mass is double the amount of mass. The hydrogen is having the mass 1, it is having the mass 2. Why the thing that mass is so high? So the kinetics changes. The kinetic like 6 to 10 times it is slower. When the kinetic isotope is there, that significantly go at the rate of metabolism in the long run. That is the main purpose of using the drug discovery program. And also, many of the cases that it is present for other drugs, in some cases the other drugs are taken and one cannot take that other drug. That the doctors will advise, no, you are taking this drug you cannot take it, that you avoid that drug. But in the case of the deuterium, one certain cases, the advantage is in present of other drugs also can use the drug without much harmful effects. The metabolites formed also very less in this case. Deuterium. Now, what is important? Replacement of hydrogen with deuterium does not have the effect of lowering toxicity. How the lowering toxicity happens is studied it, but the metabolites generally is forming. When you use the drugs are undergoing metabolism. And due to the metabolism, some of the metabolites are very much toxic in nature. So when we study the thing, since the deuterium drugs are slow in nature and metabolically stable, that is not producing any much metabolites which are toxic in nature. In the case of this one, they studied that there is no effect on biological potency and selectivity, but very much lowering the toxicity. So this advantage they have derived. Toxicity problem is very, I told you initially, is the real bottleneck in the pharma industries. This is the good solution now people have got it. So more and more people are trying to reduce the toxicity by introducing that. Now, what is the current status of your deuterium drugs? After seeing the success of tetrabenzene, I have around 20 molecules are in the pipeline or almost for the clinical trial. With 6 of them, I noted down, but the name is not known to me. I have not just written the thing. The pharma people will be knowing better, and they are reached almost the final clinical phase 3 trials. In future, within some years, I hope that some of the drugs, very important drugs, will be coming out, and much more success will be there. I can see that the success. Now, coming to the other uses, that in pharmaceutical uses are many, but I have not covered that so much thing. But that's also very important because heterogeneous crowd, because chemistry, research people, and other engineers are there. I thought slightly will balance it. Of course, the pharmacy is slightly I get more, and others use also there. Heavy water is used in groundwater hydrology also is very much useful. Previously, treated water, wherever treated water people were using it, that they are now replaced with D2O. And the geological research mapping of water also. And secondary oil recovery program, out of which secondary oil program I was involved, so I can tell something about recovery of this secondary oil recovery program. It's very important. The case of, especially we have supplied that appreciated water, the ONGC companies, they're offshore and onshore. We went at the oil wells, we have recovery program here. What is the recovery oil, the secondary oil recovery program? Primary recovery, the oil wells are there. It's how much tons of what is the oils are recovered from the oil wells. Due to the primary is with the insertion of the pipelines, what with the high pressure, the most of the oil will come. Most of the oil means more than say around idea is around 30% oil will come. Rest of this will be tapped in the matrix of the ground. So how to recover the thing? Recover the thing. First of all, they should study how much residue oil is there, how much, what is the flow, and what is the way we can recover. All the studies they undertake using treated water. Now, using the treated water, the quantity of the treated water is in liters. But in the case of the deuterium, what is also people have started using it, but it is in case of they are using 1 liter, 2 liters, we'll have to use say a maximum say 500 liters or 1 ton maximum we can use it. But the information we are getting, enormous valuable information we are getting from technology, how much quantity is trapped. That is more important than the thousands of crore they get. But we have supplied appreciated water worth of 1 crore. 1 crore they are using, when you can imagine how much the benefit they will be deriving from the appreciated water program. So the thousands of, because the oil one day shutdown, you know, the 10 to 50 crores loss will be there in the oil sector. So without this study, they cannot get the information from the this ONGC oil wells. So in that way, the deuterium can definitely can replace in future that oil secondary oil program. All the companies have started in looking into that line. They started because it is not much expected because tritium already they used it. They want to replace the thing with the deuterium. In abroad, some companies have already started using it. In future, deuterium program. This also we want to propagate in the oil sector in future. So those who are in oil sector, we can use it. In addition to that, the pipeline leakages, column scanning, all the things people are using that appreciated that the reactive things are used now because the advent of the modern spectrometer, one can utilize the DTO in all the sectors, like in all sectors, the R&D purpose, they can use it. But what is the advantage of this thing is without shutting down the plant, the leakage and the blockage can be certain with this program. I think with this, I would like to uh, end my talk and thank you at all for listening my with patience. And sorry for the trouble, but some of the disturbance happened because of the technology. It is not my fault. I hope everybody have heard about it. Now it is clear.
Yes, sir.
Now,
Thank you.
uh,
Thank you. Thank you very, very much, sir, for the motivating speech. It was indeed a great learning lessons for us. This is actually a new topic for all pharma people. Thank you. You have actually started your talk with the labeling methods, isotopic purity, chemical exchange methods. Then you have spoken about the methods. How do we actually go for deuteration? Some catalytic methods you have discussed. You have discussed how to detect the toxicity. You have also discussed about the precautions needs to be taken at the time of handling these deuterated compounds. And you have discussed more in depth about the applications, which are nuclear applications, non-nuclear applications, and under the non-nuclear applications, you have elaborated, you have actually directed us that there is a need for deuterated solvents. So our research focus should be also directed towards preparation of standards and deuterated solvents for different types of applications like NMR and mass spectrometry. You've also spoken about the food applications, like how heavy water is used in food, cosmetics. That has definitely gained our knowledge. Cosmetics, we were never knowing about the deuterium getting utilized for cosmetic purpose, for skin protection purpose. And there are many topics covered by you, and finally you have given your more directions for pharmaceuticals purpose. And since we have been working in pharmaceutical area, you have actually directed all the researchers to to go in for deuterated chemistry type of research work. Thank you so very much, sir, for such an elaborative discussion. We have a couple of questions with us,
Yeah.
and quite exciting questions actually we have.
Okay. Okay. Whatever the
Yeah.
possible, then I will try to answer your questions. If not, I will get the answers. If I am not able to instantly answer, I definitely will answer your questions. Tell me.
Uh, yeah, the first question comes from Ananta Saikia, and I'm going to club this question with Dr. K.S. Kiran. They
Okay.
are asking, can deuterium replacement of
Hmm.
a hydrogen atom in a molecule exchange deuteration action of the drug? Let me read once again.
Yeah, ready. Hello.
Yeah, 1 minute, sir. I'll just read it once again. Can deuterium replacement of hydrogen atom in a molecule enhance the duration of action of drugs? And the next question is in connection to this, can deuterium be replaced with hydrogen atom to enhance the action of drug? It's more or less the same questions. So can you just take up this question, sir?
Yeah, I will tell you. If you are replacing the CH bond, I told you this. Wherever CH bond is there, if you are replacing with deuterium, that is stronger than CH bond. Once you are using in the biologic system, most of the cases it is stable, except one or two cases that enzyme reactions or biochemical change due to the biochemical reaction unstable. But otherwise, CD bond is stronger. Therefore, it is very much used in this type of studies, tracer studies. Whereas NH bond, OH bond, they are all labile. Only CD bond is stronger and it will remain intact because for carrying out the reaction we are using catalyst for exchanging with hydrogen with deuterium. That same way, if you want to exchange with CAD to CH, we require again catalyst, which is not we are not using any catalyst for reaction in the biochemical system or in vivo or in vitro systems. So that will not say exchange will not take place.
Okay, sir. There's another question coming from Mr. Ananta. He's asking, bacteria and yeast can be adapted to grow in heavy water, but will higher plants grow at 100% level of heavy water?
The plants, no higher plants. No,
Yes,
higher
sir.
plants, higher plants, it is very difficult. Adaptation one has to study because higher plants, what is the use of the thing, we will have to think about it. Anybody can try that thing. If enormous amount of use is that people have studied with the D2O, with the uptake study they have studied where the leaf it goes, where the stem it go through stems it is, where the different parts of the plant they have studied. Only the tracer purpose it goes, but same time they have not studied the 100% of the because the costly water who will apply for the thing, and people may think that we can get the labeled compound biosynthesis, but in the case of photosynthesis 100% labeling of the we have studied with one plant, one or two plants we have studied that quite successful, it is growing. Okay, but all
Okay.
plants I don't know. With the Canada indica leaf, I will tell you Canada indica leaf
Hmm.
bio. I will tell you we have got the technology to make sugars from Canada indica leaf. Okay,
Hmm.
photosynthesis 100% D2O one can use it with can without any problem. We can synthesize the sugars using that technology. Okay.
Okay, sir. Yeah, sure. I think this addresses the question. Another question is, will substitution of deuterium for
Hmm.
hydrogen makes compound optically active that would be otherwise achiral?
No. This you are replacing the hydrogen only. No, optically active.
Mhm.
Uh, whatever the things are, that will intact. It will be there, and there is no optical activity involved in this any of the process. What process is the thing that is you have to specifically you have to tell the reaction, then you can understand.
Okay, sir. There's another question from Ms. Meenal Rane. How is the performance of deuterated water with herbal drugs?
Herbal drugs. Herbal drugs, now this is the one new area you are thinking about it. That much knowledge is not there. The herbal area, I have not come across the thing. You can also think about thing and tell me the thing. This I don't have any idea. I can tell you the answer after some time, not immediately. I have given my email at ID. So for your circulation, please circulate the email thing. Any question is there, I'll be answering. At same time, any help is required to start up for this use or produce the deuterium level one. Definitely, I'll be the catalyst for the because heavy water board and the DA is a closed system. If you want to open the system, you people should participate and come out with a program. I definitely I'll help you. And two universities I helped for the preparation of deuterium labeled compounds. In that way, any this thing is really in your mind to start using or start producing, they are welcome. And my email can be circulated to all the respected audience.
Definitely, sir. Definitely.
Okay,
So we will take one more question. Uh,
okay.
uh, one- Yeah, the question is from Dr. Harita Desai. She's saying, "Thank you, sir, for informative session. Are are there any toxicity consideration while using heavy water in cosmetic preparations and lactating mothers, which you made a mention in your talk,
Yeah, yeah.
sir?"
No, yeah, yeah. The IAEA, the International Atomic Energy Program, they have already studied using the. But if we are taking the 1 ml or 2 ml, that little, very little only used for the analysis. Okay, for the lactating mothers, they are giving an infant. They take the blood and they check the nutrition value. They check it. Okay, for that trials are limited there. They're making the protocol very limited. The ml only there. They are not. I told you once you replace 25% of body fluid. So immediately it will not exchange. If you drink water, it will eliminate also. At same time, if you continuously you are drinking, then only the things 25% is replacing is possible. Otherwise, it is not possible also. So there is no harm
Yes.
in the cosmetic preparation also. When you are applying, you are going to apply with a very small quantity. In that case, a very small amount of deuterium is there. Now, how much penetration they are studying? That's why they study the penetration thing, how it is going to the blood or anything. They study very clearly. If it is safe, only this they will release the cosmetics also. It is cosmetology also part of your pharmacy. It is comes, I think. No?
Yes,
So
sir.
that,
Yes, sir.
that safety aspects will be really they will taking care.
Sure, sir.
Otherwise, it is safe.
Now, sir, we will take another last question. Uh, this question is coming from Dr. Madhur Kulkarni. She is asking, would replacement with deuterium affect the drug receptor interaction?
Pardon? Again, it means drug interaction.
So I'll repeat the question. Would
Okay.
replacement with deuterium affect the drug receptor interaction?
No, drug interaction it is not. Hello, hello. It is
Yes,
not
sir.
affected. It is not. It is not much affecting much. Hello.
Yes, sir. You are audible. Yeah,
That
some
not much.
disturbance.
So receptor that only it is only slowing down will happen. Wherever see, I told the biochemical reaction is not much affected. That enzymatic reaction people can carry out the enzymatic reaction. Okay, the same way receptor work also one can do it. What is important is the only slowing down reaction. The slow, the kinetics will be slow. The rest of the chemical reaction, everything is will be same. As far as I'm concerned, the receptor studies also one can carry out because using the tritium receptor studies are carried out. We have supplied some drug that the glutathione series, rosiglitazone one compound we have supplied to Cadila, tritium labeled. They are used for receptor studies. So when the tritium is used, tritium is very high, 3 times mass than the proton. So this is a 2 times. So definitely the receptor studies will not be affected as far as I'm concerned because we have got the experience of supplying to Nidus Cadila long back for receptor study with tritium labeled rosiglitazone we have studied, we have given. Okay, so based on my experience, I'm telling receptor studies one can easily take up. Definitely it will work.
Yes, sir. And also, in your talk, you also made a mention about the potency of the drug is not changed. Only we are trying to reduce the toxicity by deuterating the compounds. So
Yes, yes,
definitely,
yes.
I'm sure this actually conveys the message that the drug is not going to deuterated compounds are not going to affect the receptor interactions. Thank you very, very much, sir, for spending time with us and for your valuable invaluable guidance and directions, I would say, because you have actually guided us that these are the research topic of interest. One should also focus that farmer people are actually more focusing on the regular traditional ways of research, but this is another important topic which is growing, and actually focusing more attention, okay, for better repurposing purpose. That's what in the earlier morning talk also we've had with Professor Digani. She gave us the direction that one can also use deuterium compound for repurposing purpose. So I'm sure this has given us a lot of information, and on behalf of all the participants and Bombay College of Pharmacy, I thank you very, very much for spending your time and giving us the directions and guidance. Thank you so much, sir.
Okay, thank you all, and thank you for the once again. I thank the organizers for giving this opportunity, especially Dr. Patel. Special thanks to Dr. Patel, Dr. Sagar Patel. Okay, thank you very much. Thank you.
Yes, sir.
Okay,
Thank
I
you,
can
sir.
disconnect. I can disconnect or I can disconnect.
Yes, sir. Yes,
Okay,
sir. Yes, sir.
thank you. Okay.
Over to you, Dr. Vivek and Dr. Sagar.
Yes, ma'am. Thank you, Dr. Padmanabhan, for this wonderful session. Also, Dr. Premlata for chairing the session for us. This brings us to the end of the second session for the day 2 of this FDP program. Now we have a last session for the day, which is scheduled at 2:30 p.m. So we are going to end this meeting soon, and we will again meet at 2:30 p.m. post the last lunch session. So see you all at 2:30 p.m. Stay safe.
Ready? So,